Magnetogenetics refers to a biological technique that involves the use of magnetic fields to remotely control cell activity. In most cases, magnetic stimulation is transformed into either force magnetomechanical genetics or heat magnetothermal genetics, which depends on the applied magnetic field. Therefore, cells are usually genetically modified to express ion channels that are either mechanically or thermally gated. As such, magnetogenetics is a cellular modulation method that uses a combination of techniques from magnetism and genetics to control activities of individual cells in living tissue, even within freely moving animals. This technique is comparable to optogenetics, which is the manipulation of cell behavior using light. In magnetogenetics, magnetic stimulation is used instead of light, a characteristic that allows for a less invasive, less toxic, and wireless modulation of cell activity. Cell activity control is achieved using magnetic compounds such as ferritin, a protein produced in mammalian metabolism which serves to store iron in the tissues, or magnetic nanoparticles. These compounds are designated to link the ion channels that are genetically expressed on specific cells. Control of activity is thus restricted to genetically predefined cells and performed in a spatial-temporal specific manner by magnetic stimulation. What is membrane depolarization? During depolarization, the membrane potential rapidly shifts from negative to positive as the sodium ions rush back into the cell. They add positive charge to the cell interior and change the membrane potential from negative to positive. Function. The sodium ion Na plus is an important electrolyte in neuron function and in osmoregulation between cells and the extracellular fluid. This is accomplished in all animals by Na plus or K plus negative AT pathway, an active transporter pumping ions against the gradient and sodium potassium channels. What do sodium ions do? Sodium ions play a diverse and important role in many physiological processes acting to regulate blood volume, blood pressure, osmotic equilibrium, and pH. Technique. There are many ways to utilize magnetogenetics in research. One of the main challenges of magnetogenetics is in introducing the magnetic component into a specific system, protein, or cells or regions, mouse brain region, of the organism in question. One approach involves synthesizing the fusion protein of ion channels with ion containing proteins such as ferritins to synthesize the magnetic component in the biological system itself, neurons. Another method is to use magnetic nanoparticles such as iron oxide synthesized outside of the biosystems and design the particles so that it will specifically bind to target ion channels. The working mechanism of magnetogenetics can be classified by how magnetic stimulation is mediated to activate biosystems, such as ion channels, heat-mediated magnetothermogenetics activation, and force-mediated activation. Magnetomechanical genetics are two main categories of magnetogenetics. In magnetomechanical genetics. Pulling force, magnetic tweezers, and torque force are commonly used to mechanically open ion channels in cells and in neurons. Heat mediated activation. The superparamagnetic nanocomposites generate thermal energy under alternating magnetic fields. This process is closely related. New relaxation theory of magnetic energy. Generated heat from magnetic nanocomposites is generally used for the magnetic hyperthermia. In magnetogenetics, the temperature increase caused by genetic nanocomposites activate the genetically modified neurons via gating of thermosensitive ion channel. Force-mediated activation, pulling force. In the gradient magnetic field, magnetic objects are attracted to the magnetic field region with relatively stronger magnetic force. The force of this magnetic pulling depends on the equation of force between magnets. This force traps the biomolecules on the cell membrane to make cluster of proteins or changes the conformation of proteins which initiate the cascade of cell signaling. Torque. When the magnetization axis of magnetic particles is not aligned with external magnetic field, the magnetic particles will involuntarily continue to rotate to align with the axis. This rotating motion is the origin of the magnetic torque. The torque of the magnetic particles is proportional to the magnetic moment of the particles of external magnetic fields. The magnetic particles that are bound to the target proteins generate rotating movements in the rotating or alternating magnetic field, inducing desired cell signaling or ion channel gating of mechanosensitive ion channels such as piezo1. Here's an experiment showing that when the electric coils and magnetic bars are activated, the cells generate heat. In these two heat maps, you can see the fluorescent intensity increased. Hippocampus is a complex brain structure embedded deep into temporal lobe. It has a major role in learning and memory. It is a plastic and vulnerable structure that gets damaged by a variety of stimuli. Studies have shown that it also gets affected in a variety of neurological and psychiatric disorders. Several earlier studies have shown that nerve cell proteins, which are activated by heat and mechanical pressure, can be genetically engineered so that they become sensitive to radio waves and magnetic fields by attaching them to an iron storing protein called ferritin or to inorganic paramagnetic particles. These methods represent an important advance. They have, for example, already been used to regulate blood glucose levels in mice, but involve multiple components which have to be introduced separately. The new technique builds on this earlier work and is based on a protein called TRPV4, which is sensitive to both temperature and stretching forces. These stimuli open its central pore, allowing electrical current to flow through the cell membrane. This evokes nervous impulses that travel into the spinal cord and then up to the brain.